Go ahead.
Okay, thank you, sir. So 38 now. So good evening, everyone, and I welcome to all the participants, our respected Prakash Salami Reja, all the scientists, faculty members, research scholars, and students in today's MBSI weekly research roundtable discussion. So we all know that today's topic is the bioplastic production and applications. So truly, I am very happy that today, as already our Halami sir mentioned, that today response is very good. About 40 participants have joined today's session. So let me introduce this topic within 2 or 3 minutes. So we all know that the bioplastic it represents the means one powerful intersection of, we can say, the microbiology, biotechnology, okay, and in the Indian examples, if you take, so we can take that here that the IIT Hyderabad, just I have gone to that, has developed the cost-effective PHA-based bioplastic. ISC Bangalore has also developed the biodegradable the cutlery from the cellulose and the biopolymers. And NV Green, there is one startup in the Bangalore that who has pioneered the starch-based biocarry bags. CSIR NIST
काकी में के दुकान पर जाइए ना तो देख लिया अगर
they have developed working with the microbial fermentation technology to upscale the PHB production. Okay, so just I am explaining all these, demonstrating all these examples that how India is becoming a key player in the sustainable biomaterial. So now, as we all know, that the purpose of the roundtable discussion to enhance the collaboration, to support the PhD scholar, and to share our experiences. So now I request everyone to share your experience, whether you are working on the bacterial pH pathways or any means bioplastic, bioprocess scale up, enzymatic degradation, or the field application. So again, once again, I welcome you all in this discussion on bioplastic production and applications, and I request everyone to share your experience on this particular topic. Hello, am I audible to all of you? So just
Yes,
if you have any experience or your knowledge or research insights, I request everyone to share. Just unmute yourself. Bhuwana, Dr. Barsa, Niska Das, Bhavna Soni, Desraj Deepak Kapoor, Mr. Tejas, anyone who has research experience or any knowledge or the information you have regarding this topic, you may unmute yourself. Dr. Barsa Chaudhary, from which institute? Dr. Barsa Chaudhary. Hello.
Hello.
Yes, yes. Because
Hello.
this one is the platform for interaction or collaboration. So let introduce first. Yes.
Sir, I am from PHGVP Mandal's ASC College, Shahada, District Nandurbar.
Mm-hmm.
I'm working here as a professor.
Okay. So you're working on bioplastic, madam?
No, I'm not working on bioplastic, but I'm interested in this topic. That's why I have joined.
Okay. So do you want to share any your experience or your means knowledge on bioplastic, madam?
Sir, actually, on PHB production, some of my colleagues have worked on PHB production and plastic production. And as I feel this is an interesting topic, so I have joined here.
Okay, thank you, madam.
Okay, sir.
Dr. Seema Makhaija, Mr. Tejas B. Chaudhary.
Yes, sir.
Yeah,
I'm actually a PhD scholar.
sure,
I'm
sir.
working on enzymatic pathways for the bioconversion of lignocellulose. Am I audible?
Yes, yes, you are fine audible.
Yeah. So I'm working on the enzymatic bioconversion of lignocellulose, and in my research, I'm uh, particularly focusing on the cellulose-based bioplastic production
Mm-hmm.
that are derived from the lignocellulosic biomass. So I chosen this lignocellulosic biomass because this is abundantly available and it needs a waste management strategy. So I'm doing a dual work that I'm also managing the waste, and at the same time I'm trying new ways to produce this polymer processing technique to improve, we can say, the environmental aspect. And I'm I have isolated around 17 fungal species which are producing some enzymes. Which are producing some enzymes that can hydrolyze the lignocellulose, and which can be further processed to bio-based plastic. I mean cellulose-based plastic. Yeah, so I'm in my early stage currently, but yeah, this is also one of my application.
Because from where you have selected means you have isolated the fungal species.
Yeah, so actually, the lignocellulose basically
Mm-hmm.
it is a complex polymer. Okay, it is consisting of cellulose, hemicellulose, and lignin. So I selected the fungal species, mainly isolated them from rotting wood samples.
Okay.
Yeah, so that they can be able to produce enzymes like cellulase, xylanase, manganase, lactase, lignin
Mhm.
peroxidase, likewise. So they can effectively hydrolyze the lignocellulose, and then the further work can be carried out by some other ways. So the basic mechanism is to hydrolyze the lignocellulose, and there is another process by which we can convert this lignocellulose into, we can say, cellulose-based bioplastics.
Thank you. Yes. So, do you have any question to the Mr. Tejas? So you can ask now because he's also working on this field. So if you have any suggestion to Mr. Tejas or if you have any query, you may ask now, or you can write in the chat box also. I can read from there. Okay. So there's no any question.
Second thing, I wanted to
Sure,
thank
sure.
MBSI because this is a really very nice initiative for young researchers like us.
Mhm.
We get to know. I mean, I'm really young in this field. I'm early stage researcher, and the interactive, we can say, meetings like this, these can enhance my own knowledge, and the discussions can be really very fruitful.
Sure, sir. That this one is also our objective.
Yeah.
Yes, sir.
So there is one
Good evening.
question
Good evening,
from the
sir. I'm
Nishant's. Good evening.
yeah, sir. Vipul Patel. I am from Sanjeevani College of Pharmaceutical Education and Research, Kopargaon, Shirdi.
Okay, awesome.
Glad to know that this kind of discussion happen among the research scholar and all the professor like you, sir. Just I am having a small query that
Mhm.
I don't know how many of them of our research scholar are working on cyclic acid production through fermentation technology. Being we are in specialized in formulation and development, sir, we have a little knowledge about the microbiology aspect. And
Mhm.
we got a challenge research project from state government regarding cyclic acid production through microbial engineering. So I don't know, means if anyone who is having the knowledge regarding the scale up and downstreaming for cyclic acid, this is one question, sir, from my end.
Sure. Think that.
Uh, because most of the time India is importing the cyclic acid from China. We are relying 95% of importing cyclic acid, and you know it is
Mhm.
one of the major intermediate for antiviral drug formulation. I mean antiviral API production,
Mhm.
and that costing is more than 1 lakhs. If China is generally producing this cyclic acid through anise fruit, that is star fruit, and this harvesting and this all techniques will have a more costing, would be around 1 lakh rupees per kilogram. And by few literature, we come to know that microbial genetic engineering could be helpful for improvement in production of shikimic acid that will lower down the cost price around 40 to 50,000 per kilogram. That is just I'm addressing my issue, sir. I don't know how many of our research group are belong from expertise in fermentation, upstreaming and downstreaming.
No, with us, Halami sir is here. He is expertise in the fermentation, but
Yeah.
yeah.
Yeah, exactly. It is possible to answer your question. But
Yes,
this
sir.
time actually we are looking for bioplastics. Maybe in
Yeah.
a subsequent round table, we can actually have a topic like organic acid production by
Yeah.
microorganisms, and in there are high value organic acids. Under that, we can actually discuss this. And
Okay, sir.
yes, microbial production of shikimic acid. Actually, it is process is available, which of course you can have indigenous process, changing the carbon source, economical
Okay.
production, effective
Okay.
upstream processing,
Okay.
and effective recovery, product recovery. All these things is definitely possible. But now today we are going to talk about the bioplastics
Yeah.
application.
Bioplastic. Yes, sir.
Exactly.
I'm just
This
asking out of question. Yes,
And
sir.
this bioplastic is more the biodegradable plastics actually from microorganisms. Okay. So that is what actually today's topic is of discussion. But not an issue. Any other person can actually ask any their queries are pertaining to their research in microbiology. Otherwise, it will be never ending story.
Yeah.
I hope everyone will agree for this. What
so
is
one question
the
is here for Mr. Tej. I think that which method is used for detection of lignocellulose? Mr. Tejas,
Yes,
which
sir.
method?
Yes. I was just answering that question in the chat box. So the method that I have used in my study is using FTIR. I have dissolved the lignocellulose. I mean the material, the plant material that I'm using. I'm using wheat straw and rice straw in my study. I have dissolved that. I have crushed that and converted it into a liquid sample, and I have just used FTIR spectroscopy, which gives the specific peaks that can detect whether the material have high cellulose or hemicellulose or lignin content.
Mm-hmm.
Yeah. So
Okay.
that is the process that I have used. There are several chemical methods, but I have not explored them yet.
Okay. Thank you, Mr. Tejas.
Yeah.
And one question from the Prarthana. What are possible applications of this bioplastic? How could they be environmentally friendly?
exactly actually the core topic of today's discussion, I would say?
Yes, sir.
Because once we say it is a bioplastic means produced from the living organism.
Mm-hmm.
And from the MBSI perspective, it is from microorganisms. So once we say
Yes, sir.
microorganism, even in fact, last time actually when we had microbial polyperlimer, that time I was telling like Ralstonia is there,
Mm-hmm.
Alkalinus platus is there, Bacillus is there, Rhizobia also some are actually known to produce polyhydroxy alkonate PHA or monomer or the polyhydroxy butyrate or a polyhydroxy valerate. And these are actually synthesized as inclusion bodies intracellular. When we
Yes,
grow
sir.
them,
PHA, PHP.
yes, when we grow them in a excess carbon under stationary phase, as a available source of the nutrients, you know, for those organisms. And this can actually be extracted or isolated upon the cell lysis, you know. So this is first, of course, use is the biodegradable plastics, which is actually can be a very good substitute or replacement for the existing PET or polyethylene, this one, PET, you know, because these are resistant to degrade environmentally, they will have a very long effect in the environment. And other is, of course, one can have this sutures, artificial sutures in during transplantation, organ transplantations, wrapping material we can use as a biodegradable material. So several such actually advantage here, because this can actually be degraded by the microorganism itself. Okay. And can be produced by microorganisms. It is a green synthesis, in other words. Otherwise, if you are producing by the, if you are producing, suppose, no, conventional plastics, so it is a chemical energy intensive process, the chemical process, and causes the environmental pollution. And more important is their disposal problem, landfills, what we call the white pollution. So combating white pollution is by the biodegradable plastics, or the using the microorganisms for this purpose. Now, major hindrance actually is the limited yield, then expensive process, and
Yes, sir.
you may not get the strength of the plastic like what you get with the conventional plastics, because chemical engineers are very smart to design and develop the plastic what you describe. Microorganisms are actually moving little bit slower, but then you can actually have semi-synthetic, Somebody was actually explaining about the cellulose. Cellulose actually is under use as a biodegradable plastics, semi biodegradable. So like this way we can actually use, or you can engineer the microorganisms to produce this, or you can actually transfer the microbial genes into the plants. Because one of the major problem of biodegradable plastic production is the cost of the carbon source,
Yes, sir.
But if you engineer the plants using the microbial PHB synthesis operon, then you need not to have a carbon source, because plants will be harvesting the energy through the sunlight, and then your PHB will be accumulate PHB or PHA in the leaves, right? And then that you can actually use. Only the thing is, limitation is you may have to wait for a longer time. So these are actually the,
And one more, I think, sir, that mixing with the starch means
yes, blending.
cheaper polymer.
Exactly.
That will be also means can decrease the cost.
Exactly. Now, within a PHA, polyhydroxyalkonate is a broader classification. Polyhydroxybutyrate is the specific with the butyric acid.
Yes, sir.
We have to have actually copolymer. So valeric acid, if it is there, it will have a better tensile strength, better rigidity. Yeah. So like this way, copolymer production also is a possible through
Yes, sir.
the microorganism, or through the engineering, or through changing the media compositions through the fermentation process. So even that is also possible. And in fact, last seminar I was actually explaining how we actually achieved copolymer production in by bacillus by incorporating oleic acid, right? So oleic acid can get converted into the valerate, and this valerate actually act as a precursor along with the polyhydroxybutyrate. So such copolymer actually has a very similar tensile strength as well as the rigidity, film properties like your plastic, your conventional plastic. So this is what actually the important aspect is. And then definitely I think the country like India, we need the bioplastics. We need a microbial processes to combat the white pollution. We need the microbial bioplastics to see the environmental pollution in cities, landfills. You know, most of our cities are actually heavily polluted with only the plastic bags. Now, single-use plastic is there, but that is not the answer. What you will do after that? People still are habituated actually to have a carry bag. So if it is actually now, some of these people are actually, organizations are looking to use alternative like jute bag, or cotton, right?
Mhm.
Yes. Just yesterday only, actually, CSIR has the program of sisal. Sisal thread also can be like a jute. Sisal also can be used for as a bioplastic or as a biopolymer, right? Making these fibers and bags. But we are actually looking in this discussion about the ability of microorganisms to make a polyhydroxybutyrate, polyhydroxyvalerate, polyhydroxyalkonate, copolymer producing ability, and how that information or this knowledge can come actually in the application. So that is actually the challenge for the microbiologist also, because finally the any knowledge we gain or develop, it has to have a commercial application. It has to go to the public. So that's why we are actually looking the bioplastic, what it is, the production. So once we say production, it's cost economics. And what are the different applications also? Where all this can actually be used, right? Is it okay, Tiwari ji, from my side?
Yes, sir. Means also we have to improve the public awareness, sir.
Exactly. Subsequent is the
Acceptance
public.
of the biodegradable plastics.
Yes. So we have to tell them to say no to the regular plastic or plastic bag.
Regular.
If they says it is a biodegradable, just like as you said, the public awareness. So once
Hmm.
organic food, so we prefer that organic, right? Like this way, we have to create here awareness that it is a biodegradable, so you can accept that. Yes.
But during the public awareness, the biggest challenge will be cost, sir.
Yeah, that is what actually
Because
the more research is required to
yes,
make the
sir.
process economical, right? Microbial process otherwise tends to be expensive because of their
Yes, sir.
cultivation, because of the carbon media requirement, and because of their downstream processing, right? But once it is automatized, once it is industrialized, then it will be definitely much cheaper. Just like
Yes, sir.
antibiotics.
Now the publics are using the high cost means product for
Exactly.
better health.
Yes, yes.
So they obviously they will use means bioplastic
Exactly.
in the public. So one more question from the Nissan Sinde.
Mm-hmm.
Which is the copolymer used for the tensile strength increases?
Polyhydroxybutyrate versus bar polyhydroxyvalerate.
Yes.
1:2 ratio, 2:1 ratio, 2:3 ratio. More the valerate, better is the tensile strength.
Mm.
And that has to be confirmed by the NMR.
Okay. PHBB, I think that copolymer used for.
Exactly, exactly.
So any more question or anyone want to share your research experience? What is the role of the CRISPR-Cas system in instance? So already we have discussed this one in the last session, that role of the CRISPR-Cas. So here one more question, sir. It depends on the raw material in the purification process. So which type of the material purification process will help to minimize the cost of the final microbial PHB plastic product?
Yeah, you can be trying to answer that, I think, because it is more general question.
Cost, it is dependent on the raw material in the purification process. So may I know which kind of raw materials and purification process will help to minimize the cost of the. So raw materials means here, I think that means we have to replace the means costly carbon source. That is, we can use the agriculture residues, we can use the food waste, or we can use the industrial byproducts here,
Yeah, exactly, exactly.
or we can use the rice husk or the sugar cane. So all
Sugar
these are
cane,
the means raw materials that we can use, means cheaper
sugar
one.
cane molasses.
Yeah. And for that purification process, obviously we have to scale up the production by, so more research will be required for this one. industrial waste of furniture. Yes, the answer is good.
Exactly. But as such for the downstream processing where we actually use the organic acid, even some of the microbial processes itself can be used,
Yes, sir.
like a cell wall degrading enzymes from microorganisms can be used so that it gets released, and then you separate the inclusion bodies from the cell debris, right? Otherwise, you use the organic acids, which is more actually can cause the pollution also one side, and another side is their cost of production. So you can have actually the biological process also to use for recovering the inclusion bodies or bioplastic from the microorganisms.
Yes,
Partially,
sir.
and you can actually reduce the usage of the organic solvents. Even that is also
As
possible.
already sir has mentioned that we cannot reduce the cost just by any single area. Means we have to scale up the production, we have to improve the microbial strengths, we have to blend with other polymers, and also you have to go for the efficiency downstreaming process. Then only you can reduce the cost of the bioplastic. I use the molasses as raw materials and synthetic material to the difference right there in the color of the product. Can you Hello Tabassum, can you unmute yourself and you can share your research experience? Hello Tabassum. Tabassum, please unmute yourself.
Yeah, some of the senior members are there, so we can actually
Yes,
take.
sir.
This is the field actually is actually
Apurva Deshmukh.
yes.
Yes. Good evening, everyone. I'm
Good
really
evening,
happy.
madam.
Yeah, can you hear me?
Sure, madam. Yes, madam. You are audible, madam.
Yeah. So I'm very happy to be a part of this webinar, and this initiative is great. The explanation on PhD was wonderful. Well, I was working with Praj Industries till last year, and I'm still working, but I'm as a consultant there now. And I was working on lactic acid production. So our venture was to develop polylactic acid, which is a bioplastic. Of course, there are attempts of PHA and PHB in the long run, but as of now, Praj is interested in commercializing lactic acid, which is out in the news after all. And to put forth upon the applications of PLA, well, as per my reading goes and my experience goes, it is very cheap. It is degradable under industrial settings, and it has huge applications in the field of medicine, automobiles, construction, packaging. Very much packaging is the best way in which PLA can be out in the market. So basically, as of today, the plastic consumption in India is 21,000 kilotons per annum, and which is very huge. And though there is a ban on single-use plastics, there are 19 items which are banned under the category of single-use plastics. But still, there is a lot of plastic being used openly without any legal action to it. So definitely, if you talk about plastic, we must make diligent efforts in order to make to use reusable materials, and also buy things from groceries where you have degradable materials, degradable carry bags, for example. So one thing that I want to put as an application which is not very well known in the community is, I think, in the field of medicine, we use a lot of implants, scaffolds, we use a lot of stents and everything, which has a lot of plastic component in it. So bioplastics are emerging as a very good replacement to the conventional plastics in the field of medicine, because that plastic can be readily absorbed into the body. Only if I speak about PLA, that is polylactic acid, it definitely gets readily absorbed into the body, and hence you have a very you don't need to remove that thing from the human after that organ is healed or anything. So this is a very good application that I have come across and read. So I would add, I just wanted to state that in this meeting, and I really appreciate the scientific knowledge that is shared in this meeting. Thanks a lot.
Thank you so much, madam.
Yes.
Exactly, PLA is one such a potential actually molecule being produced by lactic acid bacteria itself, and being
Yes.
polylactic acid is being used as another biodegradable plastic and has
Yes.
a lot of commercial application, and then lactic acid production by fermentation, is a well-established procedure. And what Madam actually has told that this biodegradable plastics can be used in the implantation. So very, very actually useful where we need not to take it back, in other words, So from that
Exactly.
perspective, it is actually very useful aspect. And this is how actually the importance of the biodegradable plastic is. Please continue. You can tell few more its advantages, madam.
No, this is what I came across in my latest reading that I was actually investigating for I was studying for Prad's business. But the thing is, I think the apart from external applications, its adaptability in the body is the best use of
Yeah.
bioplastic that we can talk about.
Exactly. Pras actually has some collaboration with CFTI Mysore also for
Yes,
technologies.
I would like to have a very good interaction with you, sir, and very nice to meet you in this platform.
Thank you very much. Definitely, madam.
So, dear students, if you have any query to Apurva madam, you may unmute yourself or you can write in the chat box.
There are
No, sir.
few actually questions in the chat box.
Just you can unmute yourself or write any question in the chat box.
Tiwari ji, you can read directly from the chat box, so somebody can answer also, not an issue.
Yes, sir. So one question from that issue, Priya, can algae or cyanobacteria be viable future platforms for bioplastic synthesis?
Exactly, very much valid question. And it is possible, one has to develop this process and then you need not to go on investing on the carbon source, right? Only the little bit slower process, harvesting, we don't know how it will be easier. And then like microorganism, blue-green algae actually will be cultivating in the open source, right? So contamination chances. Otherwise, from the carbon source or media optimization perspective, it will be a very actually useful. And then some of this now blue-green algae are being actually engineered for production and several such as bioactive or secondary metabolites. Now such work is there in the progress. Yes.
And one more question for the Tabassum. I used the moulages as raw materials and synthetic media too. The difference was there in the color of product. So Tabassum, can you unmute yourself?
The bosom may not be available, I think, so you can only try to answer that.
There is no any question.
Good
And one
evening,
more
sir.
question. Good
Good
evening.
evening, sir. Sir, this side Dr. Divya Tandon from HP, Himachal Pradesh.
Sure, yes, ma'am.
So I have one question. Like I am guiding my master's student for this project, bioplastic, right?
Mm-hmm.
What we have suggested, like I have 4 to 5 students who are working on this particular topic. So I have given different substrates for the production of this bioplastic. And like sir said, we are utilizing various kind of agro waste so that we can replace the expensive carbon source, right? So whenever I will see the literature, but I found that it's not that all kind of microbes they are accessible to for the appreciable bioplastic production. So what we have planned or make a flow chart, like if we procure microbes which are known to produce bioplastic and see their efficiency of producing bioplastic on different substrate. So can you suggest, is it okay or we must go for isolation?
Can you repeat your question once more?
Okay, sir. Like we are doing bioplastic production on different agro waste, right? We have selected 4 to 5 substrates for producing bioplastic. Right? So second question arises like we need to isolate microorganisms which are capable of producing bioplastic. Okay. So in MSc we don't have that much of duration that students can have a longer time to go for isolation, right? So what we are doing, we will procure from these organization centers like MTCC or NCL Pune, right? And we will check how much bioplastic or how much is its efficiency with different agro waste so that we can have comparison. So is it easy to carry out this process or we must go for isolation? This is my
No, no.
question.
I think you can carry out this project because the objective is different. In the first, your objective was to isolate bacteria from the environment who can produce the
Yes,
bioplastic.
sir.
In the
Yes,
second,
sir.
already the isolates are there, and you
Yes,
want to
sir.
work on the increase the efficiency of the bioplastic production. So I
Yes,
think both
sir.
you can carry out this.
Yes, sir.
What's
Because
your opinion, sir? Prakash Salami, sir?
Exactly. Dr. Divya must be either MSc or PhD in microbiology. So she should be able to plan the experiment for the student. You know,
Yes, sir.
getting the industrially useful organism, there are the only two sources. One is the nature, second is the
Yes.
culture collection.
yes, sir.
Culture collection are by the research institutes. Right.
Yes, sir.
So culture collection means the thing is problem is it is somebody's culture, it is not your culture.
Yes, sir. Yes, sir.
Another thing is you never know whether it will really help you or not, whether it
Yes,
produces or not.
yes,
Right.
sir.
So you get though you get a ready-made culture, but you don't get the assurance from
Yes,
that.
sir.
So if it does, then you can look, no, change several variables, and then directly start the experiment. But when you are actually making the student to isolate, that means you are telling them to start from the scratches, but
Yes, sir.
you are
So
making
that's
them
why
to
what
learn
we
something.
have done out of 5 students, 3 will go for isolation, and for comparison, we have procured some strain from that particular microbial collection center so
Yes.
that we can have comparison. That is our isolated strain as well as the strain which we procure from these centers.
Yeah, you ensure that the culture which you have procured is really producing or not.
Yes, sir.
So that is the one. Otherwise, it can always act as a reference or standard
Yes,
material. There is no doubt.
reference.
You
Yes,
know,
sir.
all our
And
micro.
also they have the time limitation, sir.
Yeah.
Yes, sir, because for dissertation, our students will get only 6 months.
6 months
That's
is a
why.
quite longer time. Not an issue. You ensure that the culture which you have taken obtained from the culture collection center is a potential authentic culture
Yes,
and
sir.
what you are actually looking for. So
Okay, okay.
having such a reference culture, there is no harm. Otherwise the isolating the biodegradable plus polymer producing culture is not that difficult if you follow the procedure properly.
Yes,
Maybe
sir.
the yield will be different, can be different, can be
Yes, sir. That's why we can do the comparison.
exactly,
One study can be done like that.
exactly.
Okay. Thank
If
you.
you are if you are extending this work a little longer, then best way is you can ask somebody if they can share or spare the culture so that you can use that as a reference culture.
Yes,
Yeah,
sir. We can do that.
exactly.
Okay, okay. Thank you, sir.
And which one more question from the Nishant, which process is used for detection of starch and other material in fruit waste? I think that iodine method, by iodine you can test the presence of starch in fruit. Nishant Sindh, which process is used for the detection of starch and other materials in fruit waste or raw material? I think that iodine solutions can be used for the detection of starch in the fruit. So any more question now? Would anyone want to share the experience? Just unmute yourself.
If senior members are there, you can ask Dr.
Yes.
Tiwari, Dr. Seema, I see. Then Dr. Prasann Kumar is also there. He has conducted.
He has joined, sir.
Yes.
I messaged him to join.
He has not joined?
I think just I have to check it.
Yes, yes.
So just 5 minutes before I. Hello.
Exactly. Otherwise, we can conclude.
Hello.
Hello.
Am I, yes, yes. Am I audible?
Yes, you are audible, sir.
My name is Desraj Deepak Kapoor. I'm also a PhD scholar in Baba Sahib Bhimrao Ambedkar University, Central University, Lucknow. So I have done actually I have extracted PHP from bacteria from the scratch. So what we have done here, we try to lower down the cost. First we just grow the bacteria in MSM, like we see the literature. From there we took the medium MSM, minimal salt medium, and we provided 2% of glucose for the bacteria. So what bacteria did? A bacteria gave a decent amount of PHP. So now the biggest challenge is to lower down the cost. So what we have done here, we employed a cow dung based medium, a modified cow dung based medium here, and by applying that, we also got a decent amount of PHP here, polyhydroxybutyrate. So after all the process, we also got the patent for it from Indian patent about the production of PHP for the process. So we actually grow the bacteria in cow dung, and we got a decent amount of PHP there.
Oh.
Yes, sir.
Thank you, Desraj, for sharing your experience.
Thank you, sir.
Dr. Seema Makhija. So Dr. Prasun sir has joined. Hello. Am I audible to you, sir? Dr. Prasun sir.
That previous student who was explaining was interesting. Actually, he should actually explain a little bit more in detail which organism, how he identified, how he has demonstrated that it produces PHB.
Dr. Desraj Deepak.
Yes.
Yes, sir. Yes, sir.
So just take an explain that which organism means bacteria we have isolated and from what source.
Actually, I have isolated several bacteria, like Bacillus. Mostly, they are the Bacillus species. So
Bacillus,
the best, yeah. So the best one is Bacillus thuringiensis. So we went for that bacteria. We actually did a different bacterium in the MSM medium first, the minimal salt medium. Then we got a different amount of PHP there. Then the best bacteria we have selected after the best amount of PHP we getting from the Bacillus thuringiensis. So we selected it and we proceeded for the cow dung also.
Deshraj, who is your guide, by the way?
Dr. Ravi Kumar Gupta.
Okay. And which department you are
From
doing
BBA.
PhD?
Yes, sir.
Which department you are working?
Department of Environmental Microbiology, BBA.
Wonderful. So Dr. Ramchandra is there, right?
Yeah. Ramchandra is there. Yes, sir. He is proctor now of the university.
Oh, wonderful. Who is the vice chancellor now?
Vice Chancellor Rajkumar Mittal, sir.
Wonderful.
New VC.
Tiwari ji.
Dr. Seema Madam, Dr. Seema Makhija, Dr. Isai Anand, Dr. Ragini, Dr. Barsa Chaudhary. Hello. Also, you can unmute and you can introduce yourself so that we can know each other. Dr. Isai Anand, Dr. Ragini.
Hello, sir.
Yes. Hello, Dr. Barsa.
Sir, shall I ask one thing? I have a query. Whatever the products produced by microorganism, like antibiotics or organic acid, like citric acid or lactic acid, these products have less commercial value as compared to the products produced by chemical synthesis method. So, sir, so what we can do means these products are produced by chemical way and they have commercial value. They are used. And what if the products produced by microorganism, their production, their recovery, it have a lot of problems. So these are not used so much like the chemical synthesis way, amino acid, vitamins, all these things. So what is the remedy for this?
Dr. Varsamata, one question to you. That's
Yes, sir.
individually, what can induce you to means consume biodegradable or the microbial products? Means individually, you are also using the chemical product. So what can induce you to use the microbial products or the biological product in place of the chemical one?
Chemical have the hazardous effects, sir, as compared to our natural products.
Mm-hmm.
But people know this till they are going towards the chemical products. Means we know the importance of biofertilizer till farmers are using chemical fertilizers.
Because those are cheaper one. Prasun sir,
Farmers are not educated, no?
yeah,
Awareness is not there, and then they are looking for much more the easier way. They are
easier and the cheaper also, sir.
cheaper and then cost benefit. You know.
Yeah.
So due to that reason, we have to actually create those awareness. That is what the sustainable agriculture is. You know, sustainable agriculture actually talks about this. So this is actually one of the other aspect of microbiologist, how we can actually involve in achieving the sustainable development goal of United Nations. And if you see among the 20 sustainable development goals, at least in 10, microbiologists actually can contribute heavily. Maybe one of
Yes.
the our roundtable discussions should be sustainable development goal and microbiology, I guess. Tiwari ji.
Sure, sir. Next session we.
Yeah. You know, sustainable development goal and then microbiology or microbiologist role in a sustainable development goal. You know, that can be actually really interesting. You know, because microorganisms are involved in nitrogen fixation, then recycling these nutrients, bioremediation, biofertilizers, you know, then organic acid production, several of these things. So
Yes, sir.
we should actually have on the similar line. Please go ahead.
Yes, sir.
So one more question. If agriculture, oh, this one is a suggestion from the you, Deepa Lakshmi. If agricultural or food waste used as efficient and sustainable feedstock for the PHA production. Yes, sir. How can we use the biotechnology to protect the environment? This is from the Tanya. So any expert if want to answer this one, how to use the biotechnology to protect the environment? Dr. Divya Tandon is here.
Yeah, I think after discussing with the bioplastics and then conventional plastic, anyone actually can answer this question.
Yes, sir. That's why.
Right? Exactly. So whatever we have discussed, that itself is the live example or actually the valid example how the bioplastic has the several advantage over the conventional plastic. I was talking about the white pollution. I was talking about the drainage problem. I was talking about the sewage problem. I was talking about the decomposing the conventional plastics, right?
Yes, sir.
There should not be any actually doubt, at least among the microbiologists, among the participants of this roundtable discussion, to answer this question.
Yes, sir.
Tiwari Saab has switched on his video. That means we should not talk more.
No, sir.
Still 32 participants are there, so it is actually a quite good moment. Actually, I have to attend the EC meeting of MBSI.
Mm-hmm. Okay. So it was postponed, sir, or?
No. 8 o'clock they told.
Okay.
But you can continue, not an issue. I will be there for some more time. you can take few questions or you can take some opinions from few of the doctors who are available.
So
Yeah.
now, Prasun sir,
Yeah, Prasun sir, we can.
are you there, sir? Prasun sir.
Yes, yes, Vikram sir. Thank you so much. Actually, other participants were having some discussion, so I did not interrupt. Yeah, it is
Mhm.
nice to see the gathering today. Quite a
Yes,
good.
sir. It was about 44 in the beginning.
Yes, sir. Yes, sir. Uh, so, two, three things that I noticed now. One question was about algae, whether algae can be used for
Algae
bioplastic
and
production.
cyanobacteria.
Yes, and cyanobacteria, yes, they can be used. Actually, there are two classes like Halami sir has already mentioned that contamination, chances of contamination is too much if we are using
Mhm.
heterotrophs. So only autotrophs are better, but they are too slow in growth. So while we know that PHA is an intracellular product, so higher the biomass, higher will be the product. And that is the one thing, and another thing will be the downstream processing cost. So these two are the challenges basically, but yes, algae can be used to produce bioplastic, and people are engineering a lot of algae. There are so many reports nowadays using algae, they can produce PHA, but the yield is again low. So if we have to
Means
break
it is
down,
lower than bacteria, sir.
yes, yes, very, very low. And we need to do a lot of genetic engineering to actually accumulate that much of PHA. And then says the we have to degrade the cell wall. It is just like plant cell wall. So cell wall is quite hard, algal cell wall. Then only we will get the bioplastic. So that process, downstream processing becomes quite costly. So that is another challenge. Right. Then one question came, I think, regarding the master's student whether to go for means isolation or we should directly procure from culture
Or
collection.
cultural correction.
Yes, yes. So for master's students who have less time, I think from culture collection, going for a culture collection would be better because isolation and characterization will take a lot of time by the time students goes away from the university.
Mhm.
So in that aspect, miss from the time wise, it is better to go for a well known or identified culture, and we
Mhm.
already have a lot of them in MTCC as well.
Yes, he procured from the MTCC several culture.
Yes, yes, yes. So that would be fine. And for screening, anyone can go for anybody can go for this Nile blue stain or Sudan black based staining, so that students can frequently do microscopy to detect the amount of PHA or lipid that is accumulated within the bacterial cell. Sudan black staining, they can do plate screening if they want to do. They can use Nile blue stain, Nile blue or Nile red, anyone. So those were the things. Yes, I was very much interested. Some fellow working in Paraj, and she is now working as a consultant, no, for PLA. So I wanted
Mm-hmm.
to initiate
Apurva
a
madam
discussion.
is there.
Yes, Apurva madam. Yes, I was glad to learn about her work. But being a researcher from a who has worked on PHA, no, there is always a debate, PLA versus PHA, whether PLA is good or PHA
Mm-hmm.
is good, because they both are claimed to be biodegradable, biological origin, biocompatible for medical applications. Also, they both are used, right? And then recently, the discussion that has happened: chemical synthesis versus biological synthesis. In that aspect, the issue is very clear actually. In chemical synthesis, most of the time we can regulate the process, and it is almost 100% surety is there that what will be the byproducts, what will be the
Mm-hmm.
byproduct of the chemical reaction. So we know that how much amount of things is going in and how much amount of things may go out, whether we are using catalyst or something like that. In biological process, especially for PHA, the monomeric composition may not be consistent. Except for
Mm-hmm.
PHB, the monomeric composition varies even with the same organism. So that makes PHA, biological production of PHA, a bit challenging if you want to regulate the quality of PHA. So production may vary a little bit, like plus minus 10%, but the
Mm-hmm.
composition also varies. So it is two challenge that PHA have if we go for biological synthesis, and we can't chemically synthesize PHA.
So which one is the future, sir? PHA or PLA?
I will always vote for PHA, sir, because my logic for going for PHA
Mhm.
is that it is a polyester, natural polyester. Means we don't
Mhm.
have to do chemical synthesis and then in because lactic acid can be produced in very cheaper quantity. Then the next step, so it is a two-step process. Now, next step will be polymerization part of the lactic acid. So
Mhm.
because it is chemical part, so production cost is lower than PHA. But if we already Chinese scientist, I think in last meeting we were discussing this. Chinese scientists have already engineered such strain that can produce very large quantity of PHA and they can go for high cell density. So again, intracellular product. So higher the biomass, higher will be the yield.
So now,
So
but I
yeah.
think that one question not can we have a contact of the experts who worked on bioplastic during course of time? If required any help, we can contact it possible. So you were working on this bioplastic, na Prasun sir?
Mm. Yes, sir.
So just share your number so that anyone can contact you. Please,
Okay.
sir.
I'm putting my email ID
Sure,
in the
sure.
chat box. Yes, sir.
So here Prasun sir has shared his email ID.
Yes, welcome, Divya ma'am.
And also Apurva madam is here working on the PLA.
Yes, Apurva ma'am can say
Prakash,
about the PLA how it is better, and because she has already researched a lot, no, so
yes.
she may give more
Prakash
insight
Salami
about
sir
it.
is here.
Yeah, I think both the discussion and interactions were quite useful from the
Hmm.
student perspective.
Divya madam, you can also contact our Prakash Salami sir.
And then some of the queries asked by faculty as well as the student
Mm-hmm.
also was quite encouraging, especially the student from Lucknow, Dr. Bhimrao Ambedkar University. Even he has told his practical experience. So that was also actually quite useful. And then the Dr. Divya, who is actually guiding our dissertation students, how she can actually go ahead, you know. So that is actually fundamental microbiology question which we always actually struggle or suffer, you know. How best actually we can choose the microorganisms, or where do you get a bacterial microbial culture for your experimental purpose? Is it the environment or a culture collection center? So culture collection center means is not yours, right? And then, but if it is environment, then it is partly it is yours. But then long process to characterize and identify and know its potentiality. So this is what actually the challenge. But as a microbiologist, I would definitely say follow the second one. If you are industry person, then follow the first one. You know.
Yes, sir.
Exactly. So let us not extend from our side. If any other questions are there, we can take up from there, or otherwise we can close.
Okay.
Or
So now,
otherwise,
sir, I
it
would like to
was quite
request you to
fruitful
put
and
your
interesting
concluding
discussion.
comments, sir. Allow me, sir.
Huh?
I request you to put the concluding comments, sir. Because this was suggested by you today, and also it was suggested by the person, sir, in the last session.
Yeah, exactly. This was, of course, in the parallel line of the session which we had last week on
Yes,
uh,
biopolymers.
biopolymers, you know. But then I felt that the biodegradable plastics is being sidelined somewhere somehow. And then this biodegradable plastic is actually the really boon for the days to come, for the sustainable develop to achieve the sustainable development goal, as well as the microbiologist who can actually contribute in a sustainable development goal. And this can be actually the real contribution from the microbiologist as well as the MBSI to make the public also aware, right? That there is a something molecule which can actually help you, where you will not see the white pollution. You know, so from that angle, when I suggested right yesterday only to Dr. Tiwari and he agreed, I was worried that he may say, sir, similar topic is already covered. So whether we will get a 40 40 articles or not, you know. But I think today's was a really a landmark.
Yes, it was
achievement. Exactly.
very engaging and
Yes,
interactive.
yes. Actually, I was busy till 7 o'clock in the other government meeting. But then when I realized that 7:30 this meeting is there, so I think I was in a time. I did not miss any other aspect. Even
You
for
are the pillar of this roundtable discussions.
yeah, exactly.
Exactly, sir.
The EC meeting actually is going on parallelly of MBSI, which was supposed to be yesterday, Monday.
Mhm.
But few members were not there, so they are postponed to today. It is going on parallelly. I will now jump to the EC meeting. But then definitely, I think this was really useful, just like rhizobia, what microorganisms does, just like a bioremediation, what pseudomonas does, just like no, any E. coli, what it does in a laboratory. The bioplastic, I think, is also another boon. Of course, which we are seeing about the genome editing, what streptococcus did to identify the CRISPR-Cas9, right? Or any other organisms which actually made the biotechnology to revolutionized, right? So this is also the bioplastic. I think we should think in a positive way. That's all from my side right at this moment. Thank you.
Thank you so much, sir. So now we come to the end of today's session. So thank you all for joining the today's session. My sincere thanks to all the experts, Prakash Alami sir, Dr. Pasun sir, Apurva madam, Dr. Divya Tandon, also the Tejas, the PhD scholar, and all the participants, research scholars and students for joining today's session. So thank you everyone.
Thank you. Thank you, everyone.
Thank you.
And all participants are requested to fill the feedback form. It is already there in
Yeah,
the
yes, yeah, several. Just fill
yes.
out this form, okay? And who have contributed, I will share the certificate to your email ID.
Sir, can we get the video recording of this?
Yes, madam, it will be shared in our MBSI YouTube channel. Not today, madam, means tomorrow or day after tomorrow we will get this one.
Sir, if we have any queries, then the email ID which is shared by just the
Prasun
Prasun
sir.
sir. Yeah, so can we ask on that email ID?
Yes,
Sure,
madam,
ma'am.
sure.
Sure. It has
Or
been
you
shared
can,
for that.
if you have any query, madam, you can put in my WhatsApp number. I can forward it to the expert.
Thank you, sir. It's
Just I have shared
unfortunately
my WhatsApp number. If you have any query, you may you may just forward your question. I will forward to the expert in any field.
I didn't get your number, sir. I didn't get your number.
Just I am sharing it here.
Okay.
8670725693.
Thank you so much, sir. Thank you so much.
Thank you everyone for joining today's session.
Thank you.
Thank you so much.
So much.
Yeah.
Thank you, sir.
Thank you, sir. Thank you so much.
Thank you, Tejas.